Hello, I hope you enjoy the field trip to Königsberg, and now the time has come to look into algorithmic ideas behind genome sequencing. We have a lot of territory to cover, but the good news is that we will be joined today by Dr. Sean Pham, one of the leading experts on genome sequencing. I wanted to start from a question. Who are these people who look like criminals on mugshots? And why they appear on the same slide with three great mathematicians from three different centuries? By the end of this lesson, you will learn the answer to this question, but some of you have already guessed that we are interested in these people because we are interested in their genomes. You can think about the human genome as a 3 billion nucleotide long book written in ACGT alphabet. And some of you may think that since humans are so involved, then their genome must be the longest genome known. This is not true. There are genomes that are hundreds times longer than the human genome. As we speak, there are thousands of genome sequencing projects that is being conducted all over the world. Biologists are interested in genome sequencing because they can learn a lot from studying the genome. For example, they can understand the function of a human gene by studying a similar gene in fly or even in bacteria. And of course, there are numerous applications of genome sequencing in medicine, agriculture, biotechnology, and many other fields. Genome sequencing started in 1977 when Walter Gilbert and Frederick Sanger invented the first DNA sequencing technologies. Usually, great discoveries wait for 2 to 3 decades before they are awarded the Nobel Prize. In this case, the Nobel Committee waited just 3 years before it awarded the Nobel Prize to Sanger and Gilbert. It was obvious that this discovery will have enormous implications for science. However, as great as this discovery was, sequencing technology invented by Sanger and Gilbert was very expensive. It would cost $3 billion to sequence the human genome with this technology. It did not stop biologists who in 1990 started Human Genome Project that is still up to this day the largest collaborative research project in biology. The goal of this project was to sequence human genome in 15 years and to deliver it to the public by the year 2005. In 1997, there was an unexpected entry into human genome sequencing race. Craig Venter founded a private company, Celera Genomics, with the stated goal to sequence the human genome ahead of public human genome project. And this was great for everybody because the race to sequence the human genome intensified, and by the year 2000, 5 years ahead of schedule, the human genome was sequenced. Immediately afterwards, biologists started sequencing many other genomes. And here you see all 9 mammalian genomes that were sequenced in the next 10 years. It looks like a great set of animals and a large number of biological sequencing problem. Obviously, biologists were very busy sequencing these genomes. However, they wanted to sequence thousands of genomes, and in the framework of existing technology, it was just not feasible. The existing technology was very expensive. And that's why the number of companies all over the world started a race to invent new next-generation sequencing technologies. They succeeded, and they reduced the cost of sequencing by orders of magnitude. As a result, we can now move from sequencing of a reference human genome to sequencing personal human genomes. Reference human genome represents an average genome of average human genome, but we want to find the differences between genomes of different humans. And this difference may appear to be small. In particular, between every 2 humans, you expect to see roughly 1 mutation in thousands nucleotide. However, these differences may be extremely important, as they are responsible for thousands of genetic diseases. Personal medicine and personalized genomics have important implications in medicine, and in 2010, Nicholas Walker has become the poster child of personalized genomics. His life was saved by genome sequencing. Poor child went through dozens of surgeries because doctors failed to diagnose his condition. Afterwards, they decided to sequence his genome and found a rare mutation in a gene linked to a defect in his immune system. As a result, immunotherapy saved the life of the child. And with human genome sequencing cost falling down, we expect that very soon the cost of sequencing the human genome will fall under $1,000 mark. As a result, sequencing genomes will probably become as routine as X-ray today. At the same time, there are numerous projects to sequence various species. And in 2010, biologists started 10,000 genome project to sequence 10,000 vertebrate species. Just think about this. A single human genome in 2000, 10,000 genomes in the year 2010. To accomplish this genomic revolution, we need to develop algorithmic techniques for sequencing genomes, and in the next segment, Dr. Sean Pham will tell you how it can be done.
Before getting into the algorithmic details of the genome assembly, allows me to introduce a crazy yet similar problem to the genome assembly problem. It is the newspaper explosion problem. We have multiple copies of the same New York Times newspaper. We put them on a pile of dynamite and trigger the explosion. But please do not do it at home. This is just hypothetical. So the explosion tears the newspaper into pieces. Some of these pieces got burnt completely. Some of these got burnt just partially. The question, what did the newspaper say? Well, in order to reconstruct the newspaper from these remaining pieces, there's a very important intuition behind. We use the overlapping information to construct the newspaper. In this picture, you see that two pieces, and we can see that they have overlapping information. So we merge these two pieces into one bigger piece and continue the process. If you are lucky, then you are going to see the newspaper, and the content of the New York Times in June the 27th is about the human genome DNA. Now, I'm ready to describe the genome sequencing and assembly in the language of newspaper explosion. Similar to the newspaper explosion problem, where we have multiple copies of the same newspaper, here we also have multiple copies of the same genome. In this slide, you may count that we have 4 copies, but in reality, we have millions of copies. Also similar to the process that the explosion tears the newspaper into pieces, we also fragment the DNA into smaller fragments. These short fragments are called reads. Some reads got disappeared. And the task is, from these remaining pieces, reconstruct the original genome. Remember, there is no position information is known. So a way to solve this puzzle is to apply the same intuition as you solve the newspaper explosion puzzle. Use the overlapping information. If you see 2 reads here, they have overlapping fragment. Then you would reconcile these 2 pieces into a single piece and move further. So this is the overall picture of the genome sequencing and assembly. You have multiple copies of the genome. The sequencing machine generates reads. And the task of the genome assembly is to, from these reads, to reconstruct the original genome. What makes genome sequencing so difficult? The genome is like a book written in the alphabet of nucleotide, ATCG, but its length is 3 billion. But unlike we read a book, a modern sequencing machine cannot read from the start to the end of this book, but instead they can only shred the genome and generate short reads. So some people think that the genome puzzle is actually a giant puzzle. But please remember, this one is not similar to the normal jigsaw puzzle, where pieces do not overlap. In the genome puzzle, because reads overlap, so this should be called an overlapping puzzle. Now, we are ready to put the genome assembly problem into the language of computer scientists. Let's try to formalize this problem. So the genome sequencing problem, input is a collection of string, output is a string genome constructed from read. Is it a well-formalized problem? Let's ask Pavel, because last time, remember, in the replication chapter, I criticized Pavel for not giving a well-defined problem. Now let's ask him. Well, this is not a computational problem, okay? So it allows me to first define what is k-mer composition of a string. Given a string, a k-mer composition of a string is a set of all k-mers in that string. So we can order the k-mers in this order according to the positions where they appear in the genome. But please note that the position information is not available. So a more correct way to order these k-mers is lexicographic order, like in the dictionary. So the string reconstruction problem, input is a collection of k-mer, output a genome such that the k-mer composition of the genome is equal to the collection of k-mer. So we have a well-defined problem, and what is next is to build up intuitions and algorithms to solve this problem. What is the most natural way to solve this? Well, let's start with TAA and assume that this is the first 3 mers of the genome. If TAA is the first k-mer of the genome, then what is the second k-mer? The second k-mer should start with AA. There's one k-mer starting with AA, that is AAT. Then what is the next k-mer? The next k-mer should start with AT. Here we have 3 AT, but they are all ATG, so we choose one. What is the next k-mer? The next k-mer should start with TG. Here we have 3 k-mers starting with TG. Which one would we choose? I don't know. Let's pick a random one. Okay, so the next k-mer should start with GT. Here we have 1 k-mer starting with GT, and the next one, TT. So the next k-mer should start with TT. But in our collection, there is no k-mer starting with TT. Well, then certainly there is a need to develop new algorithms for solving the problem. Let's now design the algorithm for solving the genome assembly problem. Let's have a string and its 3-mer composition, which is a set of all 3-mers generated from the string. If we represent each k-mer as a node, then the genome is a path through all the nodes. But can we construct this path without knowing the genome, but only from its composition? Well, yes, we can. We simply need to connect two k-mers if they overlap by k-1 mers. In this case, for example, we have TAA and AAT here. We connect by an hash because they overlap by AA. Let's try to follow this idea. So we connect all the nodes that overlap by 2 mers. Is it all? Can we construct the genome path already? No, there are more connections, because these k-mers also overlap by 2 mers. Can we still find the genome path in this graph? I can. How? This is the way. Please note that I cheated a little bit here, because these nodes are ordered according to their positions in the genome. Well, in reality, the position information is not available. So if I slightly reordered the nodes in this graph, then the task is more challenging. For example, in this case, nodes are arranged from left to right in lexicographic order. So the genome is actually a walk in this graph. Yes, and this path actually, what are the things that we are trying to find? We are trying to find the path that traverses every node exactly once. And we call a path that visits each node in a graph exactly once a Hamiltonian path in the graph. And the Hamiltonian path problem is that for a given graph, we need to find a path that visits every node in the graph exactly once. For example, let's look at this graph. This is a directed graph. Is there a Hamiltonian path in this graph? Yes, there is. But it's a little bit tricky to find. Here it is. Indeed, this is actually the game invented by William Hamilton, a great mathematician. But Hamiltonian path can also be applied for non-directed graph also, where there is no error on the edge.
Sean presented a very natural approach to fragment assembly. I, on the other hand, will present a very unnatural approach to genome assembly, and we will debate later whose approach is better. The first thing to notice is that, in fact, there are 2 Hamiltonian paths in the graph that Sean presented. This is the first one, and this is the second one. And while Sean presented his very natural approach, it's absolutely unclear how to find either of these paths. I will do something different. First, I will slightly modify the graph. Remember, in the Hamiltonian path problem, we put 3 mers into nodes of the graph. In contrary, I will represent every edge of my graph as a 3-mer. Since this almost cosmetic difference, it's not clear why do I care so much about this difference at the moment. If we label the edges of the graph, let's also try to label nodes of the graph. And how should we label the nodes of the edge TAA? Let's label the starting node by the prefix of TAA, and let's label the ending node by the suffix of TAA. As a result, we will have this graph shown below here. It's a path currently, not a graph yet. And this is our starting point, this path labeled by streamers, where edges are labeled by streamers and nodes are labeled by tumors. And now we'll do something very strange with this graph. Let's glue identically labeled nodes in the graph. In this graph, we have 3 nodes labeled AT. Let's glue them together. Let's bring 2 of them next to each other. And next, let's bring 3 of them next to each other. Let's bring them even closer to each other. And finally, they will glue in a single vertex, as shown at this slide. Now, we haven't done yet, because there are more identically labeled nodes in this graph. Here it is, 3 nodes labeled TG. Let's bring them closer together, and finally, they will be glued into the same nodes. Are we done? Not yet. There are 2 more nodes that are identically labeled. These are 2 nodes GG. Bring them closer together and glue them in a single node. The result is the graph that we will be studying in this lecture lot. It is called De Bruijn graph of a string. And the question that we first should ask, how does this graph can help us to find the genome? Where is the genome hiding in this graph? It isn't hiding at all, because we started from the genome to construct this graph. We just made some operations to make the genome somewhat elusive in this graph, but it is there. It was always there. Let's take a look. What corresponds to the genome in this graph? Of course, it is the path that we were gluing that goes like this. This, this. And as we walk along the graph, we slowly but surely build the string, the original string that was used. Let's continue doing this, What are we trying to do? Well, since we need to explain all streamers in the graph, what we are trying to do is to find a path in the graph that visits every edge streamer exactly once. Let's continue. And after we finish this, we construct actually something that we call an Eulerian path. A path that visits every edge in the graph exactly once. And now we have Eulerian path problem that is attributed to Euler, of course. Find an Eulerian path in the graph, which is a path visiting every node exactly once. And as you remember, Euler solves this problem trying to solve the 7 bridges of Königsberg problems. This is an ancient map of Königsberg, and the orange dot on this map is exactly where I took you to the field trip. Now, what Euler did, he constructed a graph by connecting 4 nodes in this graph. Each node corresponds to a district of the city by an edge if there is a corresponding bridge between these 2 districts in the city. It is an undirected graph, in difference from directed graphs that we mainly considered in this lesson, but it doesn't matter that much. The differences between directed and undirected graphs are small, and we will be mainly working with these directed graphs. And we'll let you a chance to implement whatever other problems for undirected graph. So we now have 2 problems, Eulerian path problem and Hamiltonian path problem. Do you find many differences here? And the question arises, why did I waste your time reformulating the problems that Sean already presented? What was my goal? And in the next segment, I will give the answer to this question. So you now have 2 very similar problems for genome assembly, but it turned out that these 2 problems had very different algorithmic fates. In the Eulerian path problem, we want to find a path visiting every edge of the graph exactly once. In the Hamiltonian path problem, we want to find a path visiting every node exactly once. It sounds like a small, cosmetic, and unimportant difference. Let me ask you a question. If you were given a possibility to choose between the Hamiltonian path problem and Eulerian path problem, as presented at this slide, what choice would you make? Sean?
Well, I would like to solve the Eulerian path problem because the graph on the right looks simpler and maybe it's easier to find a path that traverses all the edges in the graph.
Sean just gave a very wrong answer. It doesn't matter that one graph looks simpler than another. In real life, this graph will contain millions, if not billions, of edges. And it doesn't matter that one of them contains 2 million and another contains 1 million of edges. What matters is whether we have efficient algorithms for solving these problems. And it turned out that the different algorithmic fate of this problem is related to the fact that Euler actually found an algorithm for efficiently solving covariant path problem. But a generation of computer scientists failed to find an efficient algorithm for Hamiltonian path problem. In fact, the Hamiltonian path problem belongs to a class of so-called NP-complete problems that contain thousands of computational problems for which algorithms are still unknown. For some time, computer scientists tried to find out whether this problem can be solved in principle, or whether maybe there is not even an efficient algorithm for solving this problem. Maybe even God cannot solve this problem efficiently. But it turned out to be very difficult to prove that there is no efficient algorithm for this problem. And in fact, the Hamiltonian path problem belonged to one of 7 millennium problems of mathematics that remains open. However, what computer scientists were able to prove is that all NP-complete problems, which means thousands of problems from this class, are equivalent. Or in other words, if you solve any of this problem, you will solve the Hamiltonian path problem. And as a result, our failure to solve the Hamiltonian path problem becomes a little bit less upsetting, because we know that we failed to solve it, but we also know that thousands of computer scientists also failed to solve it. And since our chances to solve this problem are slim, from now on we will focus on the Eulerian path problem, and we'll study it in more details. So we now focus on the Eulerian path problem and try to learn more about De Bruijn graphs. So we saw that to reconstruct the genome, we need to find an Eulerian path in this graph. But some of you noticed that I cheated. I constructed the De Bruijn graph from genome, but in real life, genome is unknown. The only thing that is known is reads, or k-mers. Can we reconstruct the De Bruijn graph from genome composition rather than from genome? Well, let's try to do this. But let's first review what we have done. What we have shown is that given a genome, we can construct De Bruijn graph. What we want to do is, given genome composition or k-mer composition of the genome, we want to construct the genome. What we will show now is that given k-mer composition, we can construct De Bruijn graph and then, using Euler's insight, construct Eulerian paths in this graph. Let's start. So in the past, we started from a path representing the genome. But now genome is unknown. What is known is only composition of the genome, and we will represent this composition, every element, every 3-mer in composition, by an edge. We will now construct also nodes of this graph by representing them as 2-mers, as we did before. Now, to construct De Bruijn graph from k-mer composition, let's do exactly the same we did before. Let's just glue identically labeled nodes. For example, AA and AA are identically labeled. Let's glue them together, bring them a little bit closer, and finally glue. Let's continue with AT, the same thing, GC, continue, 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 continue again, more, more, and finally, we are getting the same path that we were getting as if genome would be known. But we haven't done yet with gluing because there are, of course, other vertices to glue. And we proceed exactly the same way as we were proceeding in the case when genome was known, and finally construct the De Bruijn graph. So what we have just shown that even in the case genome is unknown, we can construct the same De Bruijn graph as in the case the genome is known. And this is great, which means that the next step we need to address is how to find Eulerian passes in this genome. So for construction of De Bruijn graphs, the algorithm is very simple. We simply need to represent every chimer as an edge between its prefix and its suffix, and afterward glue all nodes with identical labels. It is a two-line algorithm. If you implemented it, that you have already figured out that there is a different way to look at De Bruijn graph. If we are given a collection of chimer, we can form nodes of the graph of all k-1 mers from this collection, and for each chimer from the composition, we can connect its prefix nodes with its suffix node by an edge. These are two equivalent definitions of the De Bruijn graph. And now let me explain what was De Bruijn contribution to this area and why these graphs are called De Bruijn graphs. De Bruijn graphs have great applications in biology, but De Bruijn, of course, did not know this because he was interested in purely mathematical abstract problems. He tried to solve so-called universal string problem, which is finding a circular string containing each binary chimer exactly once. Here are all 8 binary chimers. Can we find a string that contains each of them exactly once? We can. This is the string, and if you check for each binary 3 mers, you will find it appearing in the string along the circle if you travel along the circle clockwise. For example, for this 3 mer, it appears along the circle. But how can we construct the string? Is it even possible for all possible values of k? Well, the only thing we need to do, we need to represent each of these binary chimer as an edge, right? Because it should be a chimer composition of the string we are trying to find. Afterwards, we need to construct De Bruijn graph from these isolated edges, and the De Bruijn graph will look like this. And afterwards, simply look for Eulerian cycles in this graph. Cycles rather than passes, because De Bruijn was interested in finding circular strings. Can you find an Eulerian cycle in this graph? And how can it help us to find a universal string? Let's see. Let's start working here, and we immediately get insight into the first 3 elements of the string. Let's continue this way, and when we travel through the graph, the string is enlarging and enlarging and enlarging, and finally we constructed a universal string. The fact that the graph is Eulerian implies that every chimer appears exactly once in this circular string. But what about this 4 universal string constructed from all 4 mers? Or what about 10? All 10 mers? In this case, the graph will consist of thousands nodes. Does it have an Eulerian cycle? If yes, how can we find it? We'll answer this question in the next segment. And we are now ready to prove Euler's theorem that will help us to find passes visiting every edge in the graph exactly once. Actually, we will be focusing on the Eulerian cycle problem. Find a cycle that visits every edge in the graph, because Euler was interested in a walk through Königsberg that starts and ends at the same place. There is cosmetic, very small differences between finding Eulerian cycles and Eulerian passes. As soon as we learn how to find cycles, we will learn how to find passes, and vice versa. Let me start from a question. Does this graph has an Eulerian cycle? Or in other words, is this graph Eulerian? Of course not, because there is no way out of the red node. What about this graph? It's also non-Eulerian because there is no way into the red node. And what about this graph? We need to look a little bit more closer, but we will immediately find a red node here for which there is 1 incoming and 2 outgoing edge, which means that this graph cannot possibly be Eulerian, because in every Eulerian cycle, the number of times we enter into a node equals to the number of times we leave the node, and therefore the number of incoming edges in every node should be equal to the number of outgoing edges from every node. We call a graph balanced if in degree of every node equal to its out degree, or in other words, the number of incoming edges into every node equals to the number of outgoing edges. Is this graph balanced? Of course, we can check that for every node there are 2 incoming and 2 outgoing edges, but it doesn't help us yet to figure out whether there is an Eulerian cycle in this graph. And now we are ready to present Euler's theorem. We already saw that every Eulerian graph is balanced. So this graph has a chance to be Eulerian. Euler proved that every balanced graph is Eulerian. Of course, balanced and strongly connected, because if the graph is disconnected, there is no way to find an Eulerian cycle. Let's prove this theorem. And to prove this theorem, we need to recruit an ant and let it randomly walk through the graph, of course with the condition that the ant cannot visit the same edge twice. Let's start. If ant was a genius, then he would simply walk along the graph, as I show here, visited all edges of the graph, and finally return back. But an average ant wouldn't be that lucky, and it would start traveling through the graph, start walking, and eventually it would get stuck. Let's stop for a second and think at what node this ant can possibly get stuck. Can it get stuck in the middle of the graph? Presumably not, because our graph is balanced, and therefore every time the ant is entering into a node, there should be a way out of the node. This condition holds for all nodes in the graph except for one. Which one? Of course, it's the starting node of the ant, because in this starting node, the ant has already used the first outgoing edge, so it may arrive in this node and there will be no outgoing edges left. And therefore, what we learn that the ant can only get stuck at the node where it started. What can we do afterwards? The ant failed to construct the Eulerian cycle because it didn't visit all edges of the graph. But maybe, just maybe, we can start at a different node in the green cycle. Let's start at a different node at the green cycle. Also, it's not clear what difference does it make, but why shouldn't we try to start in a node in the cycle where there are still unexplored edges? For example, at this node in the cycle, there are still unexplored edges left, so let's force that to start here at this node. And in the beginning, let's tell them, first, follow the same cycle you constructed at the previous step. So then start working along the green cycle. The ant absolutely doesn't understand why he has to do this kind of silly work working along the cycles that he already constructed. But finally, he arrives to the initial node, and now he realized that actually now he can continue working because there are still unexplored edges from this node. Then continues working, and finally got stuck. And of course it got stuck at the same node where it started. What we should do today, afterwards? Then afterwards we should start at another node with still unexplored edges. Where is this node? B? Well, this is the node with still unexplored edges. Then first will be ordered to traverse the cycle that has been already constructed. This is green-blue cycle, and then starts, continue, then is not happy, but it's okay, continue, and finally arrives to the initial node. But now, then realizes that the instructions were not that stupid because there is a possibility to explore further. And we continue, and finally, then constructed the cycle that we wanted, that visits every edge of the graph exactly once. not only and constructed this cycle, we actually just came up with an algorithm for doing this, and this pseudocode described our instructions to the end, and you can implement it, and it will result in an efficient algorithm. And if we would now put an end to search De Bruijn graph constructed for universal string, in this case, just in 3 iterations, it will construct Eulerian cycle, and therefore construct a universal string. You can check that you will be able to derive universal string from this cycle.
So in this section, we are going to talk about genome assembly using read pairs. Pavel gave great lectures explaining how to construct the Bruijn graph from reads. And from the obtained De Bruijn graph, he explained how to find an Eulerian path in this graph to identify the genome. But he forgot to tell you one important thing. There can be multiple Eulerian paths in the De Bruijn graph. Even in this simple De Bruijn graph, there are 2 Eulerian paths in this graph. Here they are. And these 2 Eulerian paths correspond to 2 different strings. Which string are you going to give to biologists? The left one or the right one, or both? Please note that we sequence only one genome. Well, this ambiguity forces us to decompose the De Bruijn graph into multiple disconnected components. Each component corresponds to a non-branching path in the De Bruijn graph. Please note that while there are multiple Eulerian paths in the De Bruijn graph, all of them share the same set of non-branching paths shown here. These non-branching paths, in their turn, correspond to contigs, which are substrings in the genome. Well, ideally, biologists would like to have one contig for each assembly. Or at least, one should significantly reduce the number of contigs. To obtain this goal, they invented the technology called read pair sequencing that I'm going to talk about. Given multiple identical copies of the same genome, we now randomly cut these genomes into large, equally sized fragments of size insert length. Now, for each fragment, we generate pair of reads from both ends of each fragment. So now, instead of talking about reads individually, we are talking about pair of reads separated by a distance called insert size. So given 2 chimeras, if they are at a fixed distance d apart in the genome, we call them a paired chimeras. So the question is how to construct the genome not from the chimer composition like previously described, but from paired chimer composition. But what is paired chimer composition? Given a string, a paired chimer composition of a string is a set of all paired chimers generated from this string. Well, because we don't have space, let's represent a pair of chimer as a 2-line expression like this. Okay? We can order these paired chimers in this order, which is the order that they were generated from the genome. But please note that the position information is not available. So a correct way to represent the paired chimer composition should be in lexicographic order, like in a dictionary. So the question now is, can you construct the string given its paired chimer composition? Or let me describe formally. Input is a collection of paired chimers. Output is a string text such that the paired chimer composition of text is equal to the collection of paired chimers. How would you solve this? Let's ask De Bruijn. How would he solve this problem? Well, I think that De Bruijn would come up with the same approach that I'm going to describe right now, which is the paired Bruijn graph. Given a genome and its paired chemical composition. It's clear that the genome can be represented as a path where the edges are labeled by paired chemers, and the nodes can be labeled by pair of K-1ers. And this path, we call it genome path. But allow me to do one crazy operation with this genome path. Let me glue nodes with identical labels together. In this slide, we have two nodes with identical label, TGAT. We glue them, and this graph is called the paired Bruin graph from the genome. Now, you may guess that the next question I'm going to ask is, can you construct the paired Bruin graph? Not from the genome, but from the paired chemical compositions of the genome. Well, the answer, let's see how we solve this problem. Let's represent each paired chemers by an hash, from the paired prefix to the paired suffix. Now, let's do the same thing as the Bruin graph. We glue nodes with identical labels together. Here we glue, continue gluing, continue. What is it? This is the genome path, as if the genome was available. But it's not all. We have something more to glue. TGAT, there are two nodes with identical label. We have to glue them. We glue. This is the paired Bruin graph from the read pairs. Okay, to recap, the paired Bruin graph for a collection of paired chemers can be constructed by two operations. First, represent every paired chemers as an hash between its paired prefix and paired suffix. Second, glue all nodes with identical labels. In the paired Bruin graph, the genome is an Eulerian walk in the paired Bruin graph. But what is the point? In the de Bruin graph, we also have this property. The genome is also an Eulerian walk in the de Bruin graph. But please note that in the paired de Bruin graph, nodes are labeled by pair of K-1ers, while in the de Bruin graph, nodes are labeled by just individual K-1ers. Both of these graphs can be obtained by gluing operations on the genome path. But because of the difference in the labeling, paired Bruin graph is obtained by fewer gluings, and that makes it simpler. In this case, you can see that the paired Bruin graph is simpler than the de Bruin graph. Indeed, there's only one Eulerian path in the paired Bruin graph that allows us to uniquely construct the genome, while the de Bruin graph, we cannot construct the genome uniquely. In practice, paired Bruin graph has been applied in assemblers, and it has shown significant advantage in the resulting assembly.
We are back to Königsberg, that is now Kaliningrad, Russia. And you have now learned how to build a megacity from millions of DNA reads and how to navigate in this city to assemble a genome. And it looks like a very simple and natural city building approach only after we learn how to do this. But it took bioinformaticians 2 decades to figure out how to apply Euler's idea to fragmented sampling. In fact, transforming real and often convoluted biological problems into elegant algorithmic challenges in one of the most difficult and rewarding tasks in bioinformatics. When I walked today through Königsberg, I felt a sense of adventure about Euler's theory and how it contributed to modern biology. Yet, biologists who invented DNA sequencing have no idea about Euler's theory and how it could have contributed to their studies. And likewise, Euler worked on very abstract mathematical problem and had no idea, of course, that 3 centuries later they will contribute to modern biology. Our field trip is coming to an end. Now I will let you assemble genomes on your own using Euler's idea. As for me, it was a rainy morning in Kaliningrad, and I think I'll go for a walk with frequent stops for cappuccino. Do you think it's possible to make a walk in this city, stopping in every coffee house exactly once?